Good evening, everyone, and thank you for joining us for the first lecture of the Oxford SARS-CoV-2 lecture series. This is a weekly online webinar series that will host a wide variety of keynote speakers from around the world that are at the forefront of COVID-19 research. Our objective for the webinar is to invite brilliant minds to give talks that are research-focused and on a wide range of subjects to foster learning, inspiration, and innovation and provoke conversations that will dictate the future of healthcare. Before we transition to the lecture, I would like to briefly introduce our guest speaker, Dr. Amesh Adalja. In addition, I would like to highlight the procedure in which the viewers may submit questions for the speaker, which will be addressed towards the end of the event during a short Q&A session. This event series is hosted and organized by the Oxford University Personalised Medicine Society, a student and faculty-led organization that works to promote the field of personalized medicine within the University of Oxford. We would like to take this moment to thank the Oxford Center for Personalized Medicine, a partnership program between the Wellcome Center for Human Genetics and the St. Anne's College, University of Oxford. Throughout the year, we collaborate to hold research talks encompassing a diverse array of subject areas, interdisciplinary symposiums, and field trips for all affiliates of the university. If you are a current member of the university and would like to get involved, please register using the above displayed QR code. I'd like to highlight that membership is free, unlike many other societies at the university. If you have any additional questions about the society, please email us at oxmed@ox.ac.uk. And for today's lecture, Dr. Amesh Adalja, a senior infectious disease expert at the Johns Hopkins University Bloomberg School of Public Health, will give a talk entitled The Characteristics of Pandemic Pathogens. Dr. Adalja has produced multiple written works focused on emerging infectious disease, pandemic preparedness, and biosecurity. In addition, he has served on multiple U.S. government committees tasked with establishing innovative guidelines for treatment and healthcare reform during infectious disease emergencies. He is also a board-certified physician in internal medicine, emergency medicine, infectious diseases, and critical care medicine. He is also a fellow of the Infectious Diseases Society of America, the American College of Physicians, and the American College of Emergency Physicians. For upcoming webinars pertaining to the lecture series, we have next week, Tuesday, May 19th at 5 p.m. British Summertime, Professor Dan Baruch of Harvard Medical School and Director of the Center for Virology and Vaccine Research at the Beth Israel Deaconess Medical Center will be presenting a lecture, the title of which will be announced later this week. And we also have next week, Thursday, May 21st at 5 p.m. British Summertime, Professor Adrian Hill of the University of Oxford and Director of the Jenner Institute will be presenting a talk entitled A Rapid Vaccine Response to COVID, Progress and Prospects. To register for these events, please RSVP using the Eventbrite to ensure your spot for these lectures. To submit questions for the live Q&A at the conclusion of the event, please use the link/QR code to navigate to a Google form that will allow for you to submit questions. If you would like to submit multiple questions, please use the form to make multiple submissions. Thank you. And now, without further ado, we would like to welcome Dr. Amesh Adalja to present his talk. Please stand by as we transition to the speaker. Thank you so much.
All right, so can everybody see my slides and hear me?
Yes, I can hear you. Yes. Yep.
So thank you for this invitation to speak to you. My name is Amesh Adalja. I'm a senior scholar at the Johns Hopkins Center for Health Security. And what I'm going to talk about today is going to try to set the tone, since this is one of the first in your webinar series, about how to think about pandemics and why they occur, what might be the predictors that you can use to decide whether or not something might rise to the level of a pandemic. Because there's this narrative that this virus came out of nowhere, that this was something that was not predictable. But it clearly was predictable. And I'm going to talk about how to go about that by kind of thinking about what it takes for a pathogen to be able to cause a pandemic. So I have about 25 slides here, and then I will take as many questions as you want on this specific topic or on coronaviruses in general or whatever you may want to ask me. So when you look at the fact that a pathogen causes a pandemic, there's a certain group of characteristics that they have to have. And I'm going to try and go through some of these quickly. So the number one thing it has to have is efficient transmissibility from human to human. If you're not an efficient transmitter, so suppose you're like, for example, Ebola, where you can only transmit through blood and body fluids, that's not very efficient, and therefore is not going to be something that you see causing a pandemic. You have to have a moderate, at least a moderate fatality rate. Not necessarily has to be something very high. If something is causing everyone that gets ill to end up in a hospital, it's unlikely that you're going to be able to see that spread very, very far, because the virus or whatever it might be is not going to have access to many people. So you don't necessarily have to have something killing everybody to be able to cause a pandemic. It really just has to have a moderate or even a low fatality rate is all that we'll usually see. Very severe fatality rates probably unlikely to be able to cause a pandemic because of the transmissibility issue. Contagiousness during the incubation period. This is something that's very important, and it's the reason why influenza, for example, has become such a difficult pathogen to contain. Because for influenza, you're contagious about a day before symptoms. So if people don't know if they're sick, they're still going about their activities of daily living, they can then spread this to other people. And with coronaviruses, traditionally, we didn't think that they were contagious during the incubation period. But for this novel coronavirus, we are seeing some individuals who are contagious during the incubation period and have led to transmission events. We still don't quite understand what all the factors are that govern contagiousness during the incubation period with this coronavirus, but the fact that it exists makes it very difficult for controlling. Even if you're not contagious during the incubation period, the fact that there's so many people with an illness that only have mild symptoms and allow them to go about their activities of daily living, going to school, going to work, shopping, socializing, that is something that's very advantageous for a pathogen, because those individuals will then spread the virus. And as you look at this coronavirus, as you look at influenza, you can see that many, many people have mild illnesses yet are still contagious. And if you look at the way the testing was set up, for example, in the United States, we were not really in the early stages able to even test these mild cases, but they were still contagious. So it's not surprising that this virus got as far as it did in the United States, because we weren't testing mild illnesses. In immunologically naive populations, one of the dangers of this virus, and with any virus, is that when it faces a population that does not have any level of immunity, it can spread basically like wildfire. And that's what you're seeing with this pandemic. This coronavirus, no one really has any immunity to it. So that translates to what's called an attack rate. A very high attack rate is what we're seeing. So even if the fatality rate or the hospitalization rate is relatively low, the fact is so many people are susceptible to it. A small percentage of a big number is still a big number. So having a population that has no immunity built up against something really allows a pathogen to move quickly. Obviously, these pandemic pathogens, no vaccine or treatment, has to be able to evade the host immune response, at least to some degree. And that's basically, that's sort of how you think about a pandemic pathogen. So this is kind of the mix of ingredients that I think about. You can look on the side here on that picture. That's actually from a British researcher, or a Scottish researcher at the University of Edinburgh, just talking about the fact that more likely viruses, when you look at these types of pathogens, there's so many different types of things that can cause infections, from bacteria to viruses to fungi. And when you look at viruses, the more host type something has, the more likely it is to be able to cause a pandemic. So the fact that if you are a virus and you can infect multiple different animal species, that makes you much more likely to be able to cause a pandemic pathogen. And just to plug a report that I did on this a couple of years ago with my colleagues at the center, it's called The Characteristics of Pandemic Pathogens. If you look that report up, you'll find a lot of what I'm talking about in detail in that report. So now that we know kind of what happens, we know that we talked about the fact that this has to be efficient human-to-human transmission. What's the most efficient way for a pathogen to transmit? So what I've concluded is it's going to have to be a respiratory or droplet type of transmission. Fecal oral transmission, like we see with polio or hepatitis A, is much less likely, because sanitation can really disrupt that type of transmission. So if you look at hepatitis A rates in the United States or in other industrialized countries, they were going down even before the vaccine, because sanitation was something that could work. However, if something is spread through the respiratory droplet transmission route, much harder to intervene, because that's people talking, that's people singing, that's people just going about their daily life. So that's very hard for somebody to actually intervene on. And that's why you're seeing such widespread transmission of this coronavirus, because it's spread in the most likely way to get efficiently between a person to person. We also think about mosquitoes as well. And I think they're interesting because if you think about some of the pathogens that mosquitoes spread, especially the Aedes mosquitoes, like dengue fever, chikungunya, Zika, those get very far pretty quickly. But you have to, and they may be an exception because they do have that ability, because Aedes mosquitoes are all over the world in many places that their range has expanded to. You do see the fact that you can get explosive outbreaks, and Zika is a case in point. But again, I'll talk a little bit about why I don't think mosquito-borne illnesses can really be a pandemic in the same sense as a respiratory virus in a second. So we talked about the fact that you have to have a whole group of things. So efficient transmission and all these other characteristics. But we always focus on viruses, but is it true that viruses are the most likely to cause pandemics? Is it possible that a bacterial pathogen could cause a pandemic, or a fungus, or a protozoa, or something else? So in this report and in this project, I try to be very agnostic and just try to get back to first principles and say, what would cause a pandemic? So I wanted to go about this in an agnostic way, but if you can't really have an agnostic position eventually when you're going to start preparing. So what do I think is the most likely type of pathogen to cause a pandemic, and why? So when you look at viruses, they're very formidable. They mutate at a much higher rate than any other type of pathogen. Remember, a virus is just a piece of genetic material wrapped in protein. So it's not something that's very, structurally complicated or intricate. And they have this transmission and replication cycle that's very, very rapid. So for example, a person with hepatitis C, they have like over 1 billion viral particles per milliliter in their blood. That's really a lot of replication going on, which gives you a lot of ability to mutate, to evolve, that you don't see with bacteria or fungi or anything else. And we have nothing like a broad spectrum antiviral agent. This is technically very hard to do because, again, viruses are just a piece of protein with a piece of genetic material with protein around them. There's not much that you can attack them with that doesn't also attack your own cells, because remember, viruses use all the host cell's machinery to do what they're going to do. So they have very little unique targets, and what unique targets they have are unique to themselves. So for example, we have drugs that work only on one genotype of hepatitis C. We have drugs that work only on one strain of influenza, like they only work on influenza A, for example, not influenza B. And most of our drug design for antivirals has been done that way consciously, to try to make very specific antivirals that don't have much collateral damage to the host, because remember, again, viruses don't have that much machinery of their own, and we don't have broad spectrum antiviral agents. And we're hearing a little bit about remdesivir, which is sort of falling into that category, but it's not something that's easy to do. We have seen some bacterial infections that have been very scary in recent years. So for example, Yemen had a record cholera outbreak. We've seen plague really take off in Madagascar, although there's a lot of questions about how many of those individuals actually had plague in Madagascar. But the reason why bacteria are not in the headlines about pandemic potential, is because of that little slide there. You can see that picture. That's from Epcot Center, which is closed now in the Disney complex. But that's a little plaque honoring the discoverer of penicillin, Alexander Fleming. The fact is, the development of antibiotics basically took most bacteria off of the pandemic list, because we do have broad spectrum antibiotics. Even when we're in a crunch with trying to find a drug for a bacteria that may be resistant, we can usually piece together something that works. And the individuals who get infected with multiply drug resistant bacteria are often debilitated. It's not something that transmits very well in the general population, but more of a nursing home and hospital-based pathogen when you think about multi-drug resistant bacteria. But bacteria also, they replicate slower, they mutate less, but so they're not really anymore a threat the way they were in the time of the Black Death, for example, because of the advent of antibiotics. So fungi are something that are really interesting, because when you look at other types of organisms, particularly reptiles and amphibians, you can see fungi basically decimating those populations. What's interesting about fungi is that they have this temperature restriction. They like to infect things that are colder. And there is this hypothesis that mammals basically evolved the temperature of 37.5 degrees centigrade or 98.6 Fahrenheit as a way to escape fungi, meaning that there was kind of a mammalian filter on our evolution that allowed us to be able to avoid fungal infections. And when you do see fungal infections now in mammal species, for example, there's often these interesting things. So you might have heard about white nose disease in bats, which are mammals, but they get that disease when they're hibernating and their temperature drops. So that's really been a major restriction on fungi. There are some caveats to this. There are bacteria like Candida auris, Cryptococcus gattii that are able to infect in a widespread fashion. Candida auris is particularly scary because it is antifungal resistant, and can have a high mortality rate. But again, this is mostly preying on the immunocompromised people. Cryptococcus gattii also can infect normal hosts, but it is something that is kind of geographically limited. One thing that's interesting of an outbreak was something called exarchylum, and many of you probably have not heard of this, but it happened in the United States where there was contaminated steroids, and you were actually injecting the fungus into people's in the area of their spinal cord. That was a really specific type of event because it was actually injecting it directly into a person's body, and they had a lot of cases. Again, showing you that fungi have big barriers to causing severe disease in humans. One thing about fungi, though, is that they don't necessarily care if they kill you because they're what are called sapronotic. They just need to find something to feed on. So if they're not feeding on you, they might be feeding on something else. So there's often this idea that if a virus or a pathogen kills its host, it's not doing a good job. But that doesn't matter if you're not what it wants to eat. It can eat whatever it wants to eat. So that's something interesting about fungi and why they can decimate populations of salamanders and snakes and frogs, for example, because they don't really mind if they kill all those other people because they can eat other debris after they kill them. One of the other things I want to talk about is prions. And I know I'm talking to a primary UK audience, and you have much more experience with prions there than we have in the United States because of your struggles with mad cow disease in the early 2000s. And prions are infectious protein molecules, and we worry about them being able to cause widespread outbreaks. But even at the worst times, mad cow disease was never at a pandemic level or a pandemic threat, because its transmissibility between people was not there. It was being transmitted from cows to humans and not really going any further. There were some cases of contaminated surgical instruments, but it wasn't an efficient human transmitter. The only time we've seen prions do this and have efficient human-to-human transmission is with a disease like Kuru. And I have a picture there of a graffiti artist in Pittsburgh, where I'm from, who writes Kuru everywhere around the city. But Kuru is something that occurred in Papua New Guinea, and that was involving cannibalism. So they had very high rates of infection because cannibalism was going on, and that's really not something most humans do anymore, obviously. So it's not something that you might expect to see causing a pandemic. We definitely have seen scrapie, for example, in farm animals, as well as chronic wasting disease, and that's something that people are looking at to see, are these going to become bigger problems, chronic wasting disease specifically? But there's no evidence yet that this has the ability to cause a pandemic. Interestingly, chronic wasting disease is something where they actually had to burn down forests because there was no way to control it, which I think is very, very dramatic. Has there ever been an extinction event in a mammal species caused by an infectious disease? The answer is likely yes. And it's the Christmas Island rat there, which is pictured. And this is the only time that we've actually documented a mammalian extinction event. And it was actually caused by a protozoa, a trypanosome. And maybe many of you might have heard of trypanosomes because they cause diseases like Chagas disease or African sleeping sickness. And this was actually spread by mosquitoes, and it decimated this rat and basically rendered it extinct. But again, when you're thinking about a mosquito-borne pathogen, you can usually outrun the vector, because even if it has a wide range where it can infect people or where it can bite, you can still outrun that if you're a human and have the ability to leave. Obviously, the rat had no way to leave. And we've seen no human trypanosome disease make this kind of global catastrophic biologic level impact. People do worry about artemisinin-resistant malaria, especially if it were to spread in Africa. That could be a continent-level pandemic, or not quite a pandemic, but a continent-level epidemic if we had multiple drug-resistant malaria there. But mostly, outside of this Christmas Island rat story, protozoa are not on that list of what can cause a pathogen. So when we go to viruses, I think it's interesting to think about why they can cause pathogens and why they can cause pandemics and what gives them that ability. So at a very high level, people try to come up with rules, but what you find are more exceptions than rules. And I think it's because we don't really quite understand exactly what happens when a virus causes a pandemic. So the first type of thing is thinking about RNA versus DNA viruses. So RNA viruses are usually able to mutate much faster than DNA viruses do. So most of your pandemic threats tend to be RNA, except there is the pox viruses like smallpox or monkeypox. Those are DNA viruses, so that's an exception. But in general, usually RNA viruses. And many of these RNA viruses, I'll get a little bit technical, replicate in the cytoplasm of the cell, meaning that they're outside the nucleus of a cell. And that tends to come up as a pandemic having a pandemic potential if it replicates in the cytoplasm. And that's because to replicate in the nucleus, you have to be very tied to one species because you have to be able to get into the nucleus. So you have to be really adapted to that type of cell. And we think in general that pandemic threats are going to be diseases that can jump from one animal species to another animal species. So the fact that they're replicating in the cytoplasm means that they're not so strongly tied or adapted to a specific host, allowing that to happen. One of the other things is segmented versus non-segmented genomes. So I have there a graphic from flu, and I think flu is the best example of a segmented genome, where you basically have a lot of different genes on different segments that can shuffle themselves. So you can see that happening with the pandemic viruses that led to the flu pandemics in 1957 and 1968, where avian viruses and human viruses basically swap segments and something new came out. And that's what gives flu that advantage. Many viruses don't have that flu. And if you look at what happened in H1N1, it was basically a triple reassortant virus. And that's why flu is always going to be the top pandemic threat because of its genome. But again, it doesn't necessarily mean you have to have a segmented genome to cause a pandemic, because the SARS coronavirus 2, which is the cause of COVID-19, does not have a segmented genome. And it's something that you have to just kind of take with a grain of salt, that there are these general rules, but there's lots of exceptions to them. So if you look here, this is just showing you with what happened during 2009 H1N1. If you can look there, the multicolored screen there, the triple reassortant. So basically, it had genes from the H1N1 1918 classic North American strain of swine, as well as strains that had picked up from different viruses and from swine viruses from different parts of the world. So this was really a conglomeration effect of different viruses. But the fact that it had so much that was like the H1N1 1918 strain, the areas that are in blue in the bottom left there, is what gave our population some immunity to this virus. So if you were born in an era when flu was in an era that was dominated by the 1918 flu, so that means if you were born between 1918 and 1957, you likely had a lot of protection, and that's why that was not a very severe pandemic, only killing about 12,000 Americans. But there are more scary flu viruses out there, and one that I would like to highlight is H7N9, which is the scariest virus I think that's out there right now. This is a flu virus that has been causing outbreaks in China for some time, since around 2013. This is now in its fifth wave or so, and it's starting to show more human tropism. So right now, this virus, H7N9, can spread from poultry to humans, not very efficiently between humans. And that's the issue, is that this is not something with sustainable human-to-human transmission. If this flu virus were to get that type of transmissibility, it would be a game changer. And we're seeing it start to get those changes, but not enough yet to transmit efficiently. There's been over 1,500 cases. And in the top right-hand of the slide, you can see it says the IRAT, this matrix here. This is something the CDC puts out, trying to rank flu viruses, and both A and B, which are the most dangerous or the most worrisome, are both H7N9 strains. So this is the one that I look at. And just to give you a gauge, H7N9's mortality is around 60%. Its case fatality ratio, much, much different than what we're dealing with now. This slide just gives you a little bit of how to think about a pandemic, and I think we went over a lot of this, but the fact is that if you have this is meant for avian flu, So it's a little bit less specific. It's a little bit specific to that. So if you've got an avian flu virus and it's widespread in animal species, wild or domestic birds, humans have to get it. And the humans have to get it, and it has to be able to replicate enough in their body so that they have infectious virus so that they can spread it to other people. And then it needs to be able to spread to other humans and not result in an early termination event, meaning maybe by chance somebody gets it and they die and they don't have any contacts. And when it comes to avian flu viruses, we're around level 4 for some of those avian flu viruses. So it is something that we have to continue to watch. And I think hopefully this pandemic, one of the side effects of this pandemic, people will be much more likely to think about pandemic threats in a new way and be much more proactive than reactive. You know, some people talk about, could there be a pandemic from something that's not from this planet? And it's actually interesting because people are looking at space bacteria, that the idea that maybe there are bacterial species that are on other planets. And as we start to think about missions to Mars and returning rocks and soil from Mars, what would happen if there were bacteria there? It's important to remember, though, that anything that's well adapted to life on Mars, even if there is life on Mars, is unlikely to be adapted to life on Earth and probably will not thrive here. Although there are some worries about Antarctic bacteria, and they're looking at bacteria in Antarctica to see how well it might resemble something if we got something from Mars. And it's important to know that when we get these Mars rocks back, they're going to be handled in a BSL-4 highest level biocontainment to see what we find on them. And hopefully it's all interesting stuff and we won't find anything really dangerous, but very interesting to think about. So that's something important to remember. The other slide is talking about the fact that what I talked about before, that many people think that fungi are perfect pathogens because they don't care if they kill their host because they have this ability to feed off of other types of debris. And that's a picture there of a fire salamander, which has been basically decimated by the certain fungi. So that's what's going on there. And sometimes people call these things perfect pathogens. And there are people who do think that fungi may develop the ability to replicate at our human temperature better. And if that's the case, they may become global catastrophic biorisks. And I think Candida auris is a really good example of that. And for those who want to know more about Candida auris, I think I would recommend reading about some of the data that they've seen on how Candida auris spread around the world. One of the other things that can make make any pandemic worse are human factors. And these are some slides, some cartoons during the SARS epidemic in 2003, just to tell you exactly why people were very wary about China in an outbreak this time. And these are some pictures of people there of politicians there trying to basically muzzle people from talking about SARS. And that really allowed SARS to get out of hand because of the fact that this was something that people could not talk about, they could not report. They were putting the cases in military hospitals, not in civilian hospitals. So I think it's important to remember that human factors can magnify any pandemic threat and make it much worse. And we're seeing that happen again with this virus as well. Just a couple of things to talk about Ebola, because people always talk about this as a pandemic threat. Fluid transmitted virus, so not something that really has pandemic potential. A couple of slides there. Ebola comes from bats, just like coronaviruses do. These are some of the bats that they were suspicious it came from. And that picture there of a tree is the hollowed out tree where a small child, the first case during the 2013-2014 West African Ebola outbreak played. And that little 2-year-old was probably getting exposed to bat droppings and started a worldwide outbreak that led to over 20,000 cases. Ebola is something, though, again, it doesn't transmit very well between humans. Most of the cases start likely because of contaminated bushmeat that people are hunting, things like gorillas or chimpanzees or these African deer, that one of them is hanging upside down in that picture there. And those individuals get infected with Ebola. They then go to their village. They are invariably misdiagnosed with something like malaria. The healthcare workers get infected, and the outbreak starts to spread. And then those people die, and then they're buried. And when they're buried, the burial practices expose more people to the body fluids. So that's usually the general pathway for how an Ebola outbreak occurs. And you can see that there's many places that you can intervene. You can say, be careful when you're dealing with bushmeat. Make sure you're wearing personal protective equipment if you're butchering an animal. You could tell people if someone has a certain clinical syndrome, make sure you put them in isolation. And if somebody dies, make sure you're burying them in the proper way. All the time, personal protective equipment should be used. And that's why Ebola outbreaks are always usually stopped in their tracks, because it's so easy to intervene if you can get the population to actually follow your recommendations. But what we saw, so if you go back to, this is a picture of Casey Hickox, who was a nurse that worked with the Ebola patients and came back to the United States in 2014. And look, you can see she's there quarantined in a tent at Newark Airport because there was a faulty reading on a temperature thermometer. And I think this is just an important example of how politicians sometimes get overboard with some of these types of with a pandemic and cause a lot of disruption based on some of their policies. And you can see that this was never really a threat to the United States. And we did similar things with some of our returning travelers coming back during this coronavirus in the United States, putting them on Air Force bases. At the same time, we knew that the virus was on both sides of the Air Force base fence. So some of that public health work that had to be squandered on some of those quarantines probably could have been better repurposed to diagnostic testing and getting our hospitals ready. One of the other things I want to bring your attention to, and I know that in England and in the UK, you've had problems with the anti-vaccine movement, especially since Andrew Wakefield came from there, that the anti-vaccine movement is something that we have to really consider seriously when we talk about pandemic threats, because many of our pandemic threats, like measles, are not pandemic threats because of vaccines. And when you look at people who get measles cases, 69.5% of them are unvaccinated. So this really is a major thing to think about. And in the last year, the United States went through a record measles year, with cases in New York and New Jersey being really astronomical and the highest on record in the era of the vaccine. And I think it's going to be important as we move through this pandemic that we continue to have positive messages about vaccines, illustrating what a life-saving technology they are and what a technological marvel they are, and really beat back the drums of the anti-vaccine movement, which really is trying to drag us back to an era that nobody wants to go back to. And we're going to have a vaccine for this coronavirus, and I can guarantee you that the anti-vaccine movement will be out there trying to undermine confidence in it. The last part of this talk, I just want to talk about how to prepare for things and how to think about this. And I have this quote from Louis Pasteur there saying, "Chance favors the prepared mind." And I think that's really important. So we know that there's all these viruses that are out there. There's all these pathogens that might have pandemic potential. And I think it's really important that we get better at trying to understand when these are jumping into humans. So that might mean sampling the blood of bushmeat hunters, people who work in abattoirs, looking for what's called viral chatter, the first forays of a virus into the human species, and looking at hotspots where this might occur. And there are lots of hotspots in the developing world. But I would also say that we need to get better at doing diagnostic testing, even in our hospitals in the industrial world. Because if you walk through any hospital, you're going to find many people with sepsis or septic shock or pneumonia. And remember, this coronavirus started out as an atypical, undefined pneumonia that no one comes up with a diagnosis for. And maybe they say, this is a virus, you're going to get better, or you're not going to get better, or we're just going to give you antibiotics and you got better. We don't know exactly what caused that. We can't afford to do that anymore. And we do have diagnostic testing capacity that can find these pathogens. They're just not being used very much in hospitals. And I think it's really important that people not be satisfied with just saying, oh, this person has a virus, or this person has some kind of pneumonia. We need to know what kind of pneumonia it is, or what's causing the sepsis in these people. And I have a picture there of Captain Kirk from Star Trek holding up a tricorder, and that was the device they used on Star Trek to be able to identify what pathogens have. We're in that criteria now. We have that ability to do that. We're in that era now where we can diagnose people this way. We just don't do it. I think we need to get more embracing of these new technologies in routine care so that we can see when the first case has occurred. And you can look in the United States, for example, this coronavirus was likely spreading here long before it was noticed, at least probably back in December, because then we knew there were cases now in France around Christmas time that had not even traveled outside of France that had contracted the virus. So this was something that better diagnostic testing would have sounded the alarm a lot earlier and made us know that thinking about this as a disease that only comes from China was the wrong way to think about this, because by the time we recognized it, it was already outside of China. So, and this is just something that we see all the time. Even with flu, so in 2013 flu season, only 15% of PCR confirmed flu cases got antivirals. We're just very bad at dealing with viruses. People still give people antibiotics even when they know they're flu. So we're really bad at doing stuff with viral illnesses. And I think we need to get better as we move into an era with pandemics. Unexpected infectious deaths. So if you look at these patients, they found 50% of the patients had a pathogen identified. So I think it's really important that if someone dies unexpectedly, to look for a bacterial infection, look for a viral infection, to try and figure out what happens in these individuals, because not knowing what's causing these fulminant deaths, even if they're a one-off in some small town, is really important for our epidemiology. And like I said before, that we're really bad even with people who are in the hospital with severe sepsis, trying to exactly figure out what caused their sepsis. So it's just an important factor. And I don't think that we have to be pessimistic about all of this, because I think we're right now at around the 40-year anniversary of the eradication of smallpox. And smallpox was eradicated. The team was led by Dr. D.A. Henderson, who was the founder of the Center for Health Security in 1997. And I had the honor to be mentored by him for about 8 years or so. And I think there's no way to describe what it's like to be in the presence of a genius, but there's a picture of him getting the Presidential Medal of Freedom. And the fact that he could do this and lead a program that eradicated something that people thought was going to fail, something that Edward Jenner, who actually invented the smallpox vaccine, thought would happen, is really great. And I think that that's a good way to end this talk, thinking about how well we did with smallpox in an era when we didn't have the tools we have today because of the genius of one man. And just the last thing I wanted to say is this great quote from Thomas Jefferson to Jenner when Jenner made the vaccine. He wrote, "You have erased from the calendar of human afflictions one of its greatest. Yours is the comfortable reflection that mankind can never forget that you have lived. Future nations will know by history only that the loathsome smallpox has existed and by you has been extirpated." So I think that's a great way to think about how well we can do when we actually put our minds to solving these what are seemingly insoluble infectious disease problems. So thank you for your attention, and I'm happy to take questions in the remaining part of this.
Thank you so much, Dr. Amesh Adalja, for being here and for that wonderful talk. And now we will transition into the questions. So we have several questions here. So let's start off with one of the questions submitted. Are there any long-term effects of coronaviruses on immune cells or the capacity of the immune system to respond to new pathogens?
Sorry, can you repeat it again? Because I didn't hear the whole part of it. Or is it written in the chat?
Yeah, it's are there any long-term effects of coronaviruses on immune cells or the capacity of the immune system to respond to new pathogens?
No, there's no evidence that the coronavirus has any kind of long-term effect. I know maybe the questioner is asking about measles. Measles does have the ability to basically delete part of your immune memory. There's no evidence that coronavirus has that capacity.
Okay. The next question. Recently, COVID-19 has been reported to cause potentially fatal inflammation in young children. What is the basis for this symptom, and is it common with other viruses?
So what we think is happening, and we're in the early days of understanding this, is that for some reason, certain people's immune system sets off a chain reaction after they've been infected with the coronavirus that leads to something that looks like Kawasaki syndrome. Kawasaki syndrome is a rare disorder, but it's been well described. It's been around for a long time. And many people have suspected that there was an infectious trigger for it, but we have never been able to figure out what that infectious trigger is. So I do think that this is an idiosyncratic reaction that certain children's immune system has to the virus. And it's something that we need to study in more detail before we can definitively link the two, but it is something to watch as we get further into this pandemic.
Okay, thank you. And for the next question, in many countries, including the US, in many countries, including the US, widespread testing for SARS-CoV-2 has not yet been implemented. How can governments and healthcare professionals work to provide greater access to tests and increase testing capabilities?
So I do think this is variable across even the United States or other countries. And I think it's a key thing that we have to have in place as we move forward, because if you can't figure out who's infected, you have no way to actually protect the rest of your population by isolating them or do contact tracing. I do think that there were really big missteps, especially in the United States in the early part of this pandemic, with how they rolled out testing. But what we need to do is basically have the ability we really have to scale up testing, and it's going to be hard in resource poor areas where they can't test for many things already. But what we have to do is remove all barriers to testing. We have to really make that a priority for manufacturers, and people are going to have to find sustainable funding for these tests and remove all bureaucratic wrangling. I think the fact that we're getting more simple tests, for example, over the weekend, there was an emergency use authorization for an antigen test made. An antigen test is much more simpler, more like a test that we do, for example, to test someone with a sore throat, looking for streptococcal sore throat. Those types of simplified tests that can be done by individuals without any training, that's what needs to happen. We need to be able to have home testing where individuals can test themselves for disease, just like they can do in the United States for HIV. We need to have that type of a paradigm push forward, but it's going to be a big challenge, especially in the developing part of the world.
Thank you. And then for the next question, there's much discussion surrounding the threat from cases which are mild or asymptomatic. Can you discuss the implications of those cases on herd immunity? It would seem like a prevalence of cases which never drive someone to seek medical care are those which would be necessary to reach herd immunity and would allow a targeted protection of vulnerable populations disproportionately impacted by the virus.
Sure. So I do think that herd immunity will take what we get. So if there are mild cases that are occurring that aren't coming to light, that's going to be useful as we try to approach a herd immunity threshold. However, even in the most hardest hit areas of our hotspots, like New York City, for example, the prevalence rate is maybe even 20% in some of the sero surveys they've done. So we're not quite near herd immunity, where we probably need to be around 60%. So to get from 20% to get from lower than 20%, if you think of the United States or any country as a whole, up to 60% is going to be an enormous cost in terms of deaths. And I think it's going to be something that you have to have a societal discussion about. And the fact is, to get to that herd immunity, you're going to put a lot of people in the hospital, and those individuals that are in the hospital are going to be taking up a lot of resources, and many places don't have the hospital capacity to deal with that. So I do think that there has been discussion about herd immunity, and I don't think it's all crazy talk about herd immunity. It's just that in order to do that, you have to be sure that A, that your society is going to accept that level of deaths, and B, that you have the hospital capacity to deal with it. And because it's going to take some time to get to that level. So I think that, and the other part was about protecting vulnerable populations. I think that's going to be key as we move forward, but you have to remember that it's not ironclad, because people in those the vulnerable populations are pretty big in most countries, and they're going to be people that they're going to need resources, and they're going to have to interact with people that are not vulnerable. So you're going to have this kind of bridge between people that you have to really be mindful about trying to keep separate, and it's very, very challenging to do so.
Okay, thank you. And then for the next question, we had one of the responses that said, in my work in Russia, I was involved in a very interesting conversation between US and Russian specialists in development of BSL-5 facilities in both countries for receipt of Mars samples where the potential of pathogen or mechanism of transmission would be considered to be unknowable. Have these programs now been canceled so that it is now standard to assume that BSL-4 is sufficient?
The questioner probably knows more than I do about this, that sounds like, but my understanding was it was going to be BSL-4. I had not heard anything about BSL-5, but that may be something that I'm not party to. I know that I would just say that specimens coming back from Mars are going to be handled with the highest level of biosecurity, whether that's the same as BSL-4 or it's something even more higher than that. I don't know the answer to that, and the questioner may know more than I do about that. So I will defer to the questioner about that.
Thank you. And then for the next question, do you think COVID-19 is present as multiple strains of coronavirus with different profiles of pathogenicity? Could this explain the diverse reactions to the virus, where some are in critical condition, whereas others simply present as asymptomatic?
No, I don't think that there's any evidence that there is different functional strains of the virus. There clearly has been separation of the virus based on certain mutations that kind of characterize it from coming from Europe or coming from China, and that's to be expected because all viruses do mutate. But there hasn't been any evidence that any of these mutations have changed the pathogenicity or the characteristics or the attributes of the virus. It's something we need to watch for, but I think what really explains how a person's clinical course is their comorbidities. Are they older? Do they have other medical conditions? Are they obese? And idiosyncrasies in their own immune system, maybe also how much of a dose that they were exposed to when they got infected, and how their immune system fared with that dose play a part. But I don't think that there's any evidence at this point to support any kind of mutation changing the behavior of the virus.
Thank you. And then for the next question, one of the responses said, you mentioned that cases likely existed in the US and there's evidence that they existed in France back in December. Can you elaborate on this?
Sure. So you remember we talked about what a characteristic of a pandemic pathogen is: an efficiently spreading respiratory virus. And we knew that this was spreading in China. So the very first cluster of cases that we heard about that were published in the Lancet, the patient 0 in that case series got ill on December 1st. So if he got ill on December 1st, that meant he was infected in November, because there's an incubation period that ranges from about up to 14 days, but at least 5 or 6 days. So he was infected in November. That patient had not been to the market that was implicated in this outbreak. So that tells you that this was spreading in the community sometime in November. And we now have backdated the first case to around November 17th, at least the first reported case in China. So that tells you that this virus was spreading for probably 2 months before it was noticed. The Chinese made their declaration to the World Health Organization on December 31st. So for about a month and a half, this virus was spreading, and it was, we know all the stuff we know about this virus, and then people were traveling to lots of different places, and this was the midst of our flu and cold season. So there was a likelihood that these cases got out of China and into other places of the country, and other countries, and were mixed into their cold and flu season and not diagnosed. And what we've seen with France is that there was an individual who presented to the emergency department coughing up some blood around December 27th, I think, if I got the date right. And he had not traveled outside of France. His child had also been sick with a similar type of syndrome, and that child had not also traveled outside of France. So that tells you that there were likely travelers coming from the epicenter to places like France beforehand, and there was community spread going on in France in late December. In the United States, our first case was diagnosed in mid-January, was a traveler from Wuhan. That was not necessarily the first case, though. We've likely missed many cases because we weren't screening people who didn't come from China. And we know that our first death has now been backdated in the United States to February 6th in California, and that person did not travel anywhere. So that person died on February 6th, meaning that there was community spread going on in the United States in January. So I do think that this likely was present in the United States as early as late December. There's no reason to believe it wasn't here if it was in France at that period of time. And I think we'll eventually be testing specimens and samples. People are doing that in Miami, for example, trying to see if anybody tests positive from earlier specimens. But I do think that we will see that this virus was here long before we suspected it was here. And that should have really changed the way we responded to it, because it was already here. It was not something that we were trying to prevent the importation of. It was already in the countries when we started our response efforts.
Okay, thank you so much. And then for the final question, why is it common for viruses and pathogens to cause a loss of sense of smell and taste?
So this is a respiratory virus, and we don't quite understand. There have been reports of this coronavirus causing loss of taste and smell, and actually 2 of my friends from New York City both lost their taste and smell and are antibody positive for this virus. What likely happens is when the virus infects that area in your nasal passages, it may be that they may be damaging the nerves themselves, or they may be in concert with the immune system causing enough inflammation to decrease their function. And my friends have gotten ill, have gotten their symptoms, the symptoms have resolved over time. But I think it's something that we don't know the complete answer to, but it likely has to do with the site of infection and the inflammation that occurs there.
Thank you so much. And I think that's the last question of the series for this event. Thank you so much for being here and for presenting your talk. And I hope everyone enjoyed the event, and looking forward to hosting the future lectures.
Yeah, thank you again for having me, and feel free to email me or find me on social media if you have any questions that I didn't get to or something occurs to you.
Okay, thank you all so much, and thank you so much, Dr. Amesh Adalja, for being here today. Thank you.
All right, bye.
All right, thank you. Bye.